Okay, so remember we were just talking about adapter proteins and these pattern recognition receptors, like the toll-like receptor proteins. And remember that they could bind to a variety of proteins, but the most common of these things is going to be a thing called MyD88, which sometimes has a protein called MAL attached to it. And the other one that sometimes gets used is TRIF. And you get TRIF on its own, or TRIF and TRAM. These are all very similar looking proteins that have similar but slightly different functions. Okay, so remember here is our toll-like receptor. And so we've got two copies here because they all are going to pair up like that. And then down here at the bottom, so this is the TIR domain, T-I-R domain. And that's what pairs these two things together. And that is how other proteins are going to stick to the TIR domains. They're going to have their own TIR domain, and two TIRs will stick together. Boom, just like that. And see how they fit together so nicely in their little drawing. It's very good. I like making the drawings. It helps me remember a little bit more. So my D88 is the inner, like main receptor, if you like. And MAL is sometimes there, and it'll kind of change the shape of my D88 and the way that it works. But my D88 is the one that's actually going to pass on the message. And so let's learn the message. So after my D88, then let's just follow this particular one. We're going to bind something like one of the IRAK proteins, IRAK. And so we're going to simplify this. We're going to take out a lot of the protein subunits, for example. But I need you to know the process. I need you to be able to go through the process and actually name the different parts. So let's just call these the IRAK proteins, IRAK. And it is 1 and 4. Remember that if you like, but just my D88 goes to Iraq. You can think of this as like the bone song, you know, the knee bone connects to the shin bone or whatever. Yeah, it's kind of like that. And this is how the message is actually going to get passed. So there are only a couple of cases where we're actually going to cover the way the message gets passed, but this is one of them. And so the IRAK proteins are then going to bind to a 3-part system. And the 3-part system's job is to bind ubiquitin and make a chain of ubiquitins. So ubiquitin is actually put on by these things called an E1, E2, and E3 ligase. I think the IRAK proteins are going to act as the E1, and then the E2 and the E3 are going to bind to them. So we don't have to remember which exact version of an E2 or E3 ligase we're actually going to activate, but we need to remember that we have activated an E2 and an E3 ligase. And so then the next thing that happens is that we're actually going to put a set of ubiquitins. So these little triangles here are ubiquitin molecules, and there's the word ubiquitin. It's named this, it's a little tag, actually a little piece of protein that gets attached to other proteins. And it's like a marker. But depending on whether you have just 1 or 2 ubiquitins or a big long chain, and depending on which amino acid the ubiquitins are going to be attached to, it can have different meanings. So one common meaning is that we put ubiquitin on proteins when we are ready to recycle these, when we're going to break them down back to amino acids and reuse those to make some other protein. That can happen sometimes, but in this case we're making a big long ubiquitin chain of a specific type that it's not going to have this function, at least not right away. It's going to have a different function. So we've gone from our toll-like receptor to the TIR domain, to MyD88, which also has a TIR domain, to the IRAK proteins, to then our E3, E2, and eventually E1 ligase that they don't even show, in which case we're going to add ubiquitin onto our E3 ligase. Okay, and this is then going to call in, or rather the E3 ligase is going to make the ubiquitin chain, that's right. This is then going to call in the protein called IKK, and it's a complex of several things. One of the parts of this is called NEMO, and it stands for something. And you know what? You can go and learn that if you're doing immunology for a living, but for now, I don't think we need to. It's got alpha, beta, and gamma subunits. We don't need to memorize that. We just need to know that it's IKK. So IKK is a regulator of a thing called NF-kappa B. And it's nuclear factor type kappa B, if I remember right. And I kappa B is a protein that inhibits it. It like slots in there and it keeps NF kappa B from going to the nucleus, which is like its only job, but I kappa B keeps it from doing its job. So again, I kappa B is like a safety mechanism. NF kappa B can't work until you get rid of I kappa B. I kappa B you can't get rid of until you have put a phosphate on it. And the thing that puts phosphates onto I kappa B is IKK. It's the I kappa B kinase, IKK. That's why it's got that name. And kinase is our generic word for a protein that adds a phosphate molecule. So that's PO4, just phosphorus and 4 oxygen molecules. It's going to add that probably from an ATP onto another protein. So that's what kinases do. They break an ATP or a GTP sometimes and take the last phosphate on there and use it to stick to another protein. And these phosphates act as like on and off switches for proteins. They change the surface of the protein and change what it's able to bind. So they kind of change the surface, and the charge distribution on the surface of a protein. So they'll make it bind to something or not bind to something. So in this case, we have our toll-like receptor, which binds to another copy of itself. So 2 toll-like receptors come together. They're going to bind to MyD88, with or without MAL, depending on which one we're talking about. MyD88 is going to bind to our Iraq brothers, Iraq1 and Iraq4, maybe the Iraq sisters. I don't know, why not? Those guys are going to bring in our E3 and E2 ligases. Our E3 and E2 ligases are going to attach a ubiquitin to a protein called TAC1. And so let's put TAC1 in here, which also gets phosphorylated by the Iraq proteins, and that sort of helps activate it. And so, all right, TAC1 bound to our ubiquitin ligases. The ubiquitin ligases can now leave, and the ubiquitin chain will stay on TAC1 because it's actually attached to that. TAC1 is then going, or rather the ubiquitin chain plus TAC1 is going to bind to IKK, and we're just going to assume that the whole molecule is here together, so we'll just call it IKK. And TAC1 is actually going to transfer a phosphate onto IKK. And so that's going to activate IKK. So it's going to be IKK with a phosphate. And these little reddish pink dots here, those are the phosphate molecules. That's how we're showing them. And it'll be that way throughout all the illustrations in this book in Janeway. So once IKK is turned on, it's activated with that phosphate. IKK is then going to put a phosphate on I kappa B, the inhibitor of NF kappa B. And once there's a phosphate on that, it's going to pop out, and so NF kappa B can be free to go to the nucleus because it is the nuclear factor kappa B. And so it's going to go into the nucleus, bind to the DNA at just the right spot to start making cytokines. So this is how cytokines are going to get made, and then cytokines are going to signal through those toll-like receptors. Once I kappa B is phosphorylated, it's actually marked for degradation, and it breaks down really fast, and that's what frees up NF kappa B to go into the nucleus. So that is the process. That is how a TLR can signal through MyD88. And if I ask this as a question, that's the form the question will take. Tell me about the signaling from a toll-like receptor via MyD88. And I might pick a particular toll-like receptor, like let's say TLR8, that does signal through MyD88. And so this is the process. This is going to be the process. Now remember, toll-like receptors can also signal through some other molecules. So TLR7, this is a shorter deal. It's going to signal through MyD88, just like that, which is going to bind to the Iraq brothers or Iraq sisters, Iraq1 and Iraq4. And then those, remember, are kinases. And so they are going to, instead of putting the phosphate onto this molecule, TAC1, they're going to put a phosphate onto a thing called IRF7. And that's something they can do if you don't have MAL around binding to MyD88. And IRF7 and IRF3 are 2 proteins. They're called interferon response factors. These are going to set off a generic inflammatory response. So these are good little messengers that will tell the cell that there's danger and we need to make some genes. And so one of the genes we'll make is going to be a thing called type 1 interferon, which is going to be like interferon alpha or interferon beta. Those are type 1. I think interferon kappa is also a type 1 interferon. Okay, and so TLR3, toll-like receptor number 3, that's fine. This is also going to be an endosome on the inside of the cell. So we knew that already from our table, right? And so we'll have, here's our toll-like receptor. Here's our TRIF protein, which again is going to have a TIR-like domain, I believe, in the tail end of it. And the TRIF protein, TRIF is going to bind to TRAF. TRAF is then going to call in are ubiquitins. And so TRAF is going to be one of these ubiquitin ligases, and it's going to put a polyubiquitin chain on a different version of IKK. So let's see. Let's call this the tank version of IKK. So you can call it IKK tank. And it's got some of the same components. The NEMO is one of the components. And we have an IKK epsilon instead of IKK beta and gamma, which were the two on the main IKK form. So call this, IKK tank version. Or maybe let's just say tank. Let's just say we attach it to tank, because that's easier to remember. That's more distinctive. So yeah, let's do that. Okay, we're going to get polyubiquitin attached to tank. That is going to lead to phosphorylation, adding a phosphate to IRF3. And when IRF3 or IRF7 gets a phosphate on it, then boom, it's going to go right into the nucleus, and it is then going to start to make messages. So these are the other ways. These three pathways, MyD88 and MAL, are going to signal through our E3 ligase, which is going to phosphorylate TAC1, and it's the IRAC proteins that are going to phosphorylate that. And then that is going to phosphorylate IKK complex, which is going to phosphorylate I kappa B, which is going to let NF kappa B go to the nucleus. Or you're going to have the shortened versions of that, which is going to be, that you have your signaling through your TLR, and it's going to be a homodimer in this case, or homodimer in this case, 2 copies, same protein, homodimer. And it's going to signal either through TRIF, if it's something like TLR3, and TRIF is going to bind to TRAF, so TRIF and TRAF go together, and that's nice and easy. TRAF is then going to assemble our polyubiquitin chain, so we're making the same thing, but via a different protein that's doing this. And there are actually a lot of proteins inside of your cells that are capable of assembling ubiquitin into chains. And so we're going to put this ubiquitin onto the complex that includes tank, so just call it the tank complex. The tank complex has got a couple of kinases in there that are going to phosphorylate IRF3, and IRF3 is going to go into the nucleus and say, let's make type 1 interferon, alpha or beta interferon. And TLR7 is going to signal through MyD88, which again is going to bind that TIR domain, which is going to signal through IRAC, which is then going to directly phosphorylate IRF7. And this can happen because there's no competition, because we don't have that MAL protein here. So we're not bringing in all the other factors, those E2 and E3 ligases, and so we just directly phosphorylate. IRF7 goes to the nucleus and done. So these MyD88 only ones, like TLR7 or TLR8, those are probably the easiest pathway to remember. The TLR3 is the second easiest. And something like TLR4 can actually signal through MyD88 and MAL, or it can signal through TRIF and TRAF, or through TRIF and TRAM. Yeah, so that's kind of neat. All right. With that, we are done with toll-like receptors. And so let's pause. That's a good place to pause this segment. And if you're doing this in 2 one-weekly installations, I would say this is a good place to end week 1. Go back and make your table, learn those things, and then draw out by hand, I would recommend, the little pathway. Make the little molecules and, draw out the pathways for TLR signaling through MyD88 and MAL, or through TRIF, or through just MyD88. And how do we end up making a message that goes to the nucleus that causes a gene transcription change? That's what we need to be able to explain. So thank you.